Okay, this is the voice recording for doing our measurements for the Carex project for Morton Arboretum. I'm just going to go through some of the major vocab that's required for you to understand. This should match up with your protocol that you're being given by your teacher. Make sure that you have a copy of that protocol when you're going through any measurements. And of course, if you have any questions, please make sure you ask them. A lab group will be a very good resource for you as you make these measurements. Make sure that as you go through any of the measurements that other people can confirm that you're at around the same millimeter measurement, because this all does go into the database and will be used not only for yourself but for other scientists as well. So we're working with the sedges, and that's the genus Carex. These are not grasses. Grasses are a little different. Grasses are Poaceae, and sedges are in the Cyperaceae. So this is one of your quick IDs for you if you're walking outside. A lot of the grasses that you'll come across in your yard are truly grasses, and they're not sedges, although there are sedges sometimes intermixed within your grasses. Grasses tend to have joints in their bodies as they reach up towards the sky, whereas sedges have these edges. You can kind of see it in this image here. They have grooves that run down them that are deep V-shaped. So we're going to go through, obviously, the sedges. We're going to look at the Carex group. So this is a plate drawn out showing some of the different species of Carex. You can find a lot of this, and I'm sure your teacher will show you, as well as Dr. Hip, as we go through a lot of the Carex Morphology project. This is one of the ones that we're going to work with in class as a practice. This is Carex grayi, and it is a little different than the one that we're going to be measuring as far as branch in the lineage component. But it'll be a nice one that will show us a lot of the traits that we need for measuring and getting you through some of the vocab components. These are the types of specimens that we will be using for recordings, and you'll have a lot of valuable information on each one of these. I just took this real quick to just kind of show you some of this before you get it in hand. So you'll all be getting an herbarium specimen when they bring these in. And it's very important that you're delicate. They are not reproducible, so if you damage them, that's the only one that they have. So we need to make sure that we are very cautious and following normal lab protocol when we're going through all this stuff. So all of these will have special ID numbers. There's also some other information down lower on it that gives you the species, some of the longitude, latitude stuff. You'll be all recording this into your data pages. This is a little different specimen, which you can see there's going to be diversity not only in the mounting, right? So there's a little less plant material here compared to this one where there's a lot of plant material. You actually see a lot of the root structure in the species. And that information that I was talking about is up on here, so you can actually see when it's first collected, the name of it. And in some of them, the name actually changes as they gain more information over time. We'll talk about how and what name you're actually supposed to record it as. It's always the most recent dated name, but we'll walk you through some of that stuff as we're actually in lab. All right, so let's talk about some of the vocab that goes with this. One of the first things that you're going to measure is what's called the culm. The culm is the piece that actually holds the fruiting body or the reproductive parts of the plant. So you look at this picture from a distance, and we see a lot of green things sticking up. Green usually represents the chlorophyll, but there's one that holds the reproductive parts, and there's the ones that are doing most of the food production and photosynthesis for the plant. So as you can see here, there's little spikes up here, and then these don't have any spikes at all. So the ones that actually hold the spikes, they have a long, slender projection, and that's called the culm. So we're going to try to get the width of the culm. So the hardest part is actually tracing back from the inflorescence, which I'll talk about with inflorescence here in a second, down to the base where the leaf sheath is. So I have a zoomed in picture here. Here it's showing you the culm, and here's a leaf blade. And so the two of them kind of come together. We want to get the maximum width of the culm, but not taking into account the blade, because that's going to be a separate measurement. So you look at the sample, it might be pretty easy to trace down the culm to where the blade meets. But in a sample like this, the problem might be a little bit more painful, so to speak. So the way to work around this is obviously you'll have these specimens. You'll have to be careful, but you'll also be able to kind of manipulate them and move them a little bit. And like I said, worst case scenario, you always ask myself or any of the Morton Arboretum staff that is with us as we're collecting these data pieces in class. So that's measuring the width of the comb. The next thing we're also going to measure is blade width. Now, blade is a green piece that doesn't have the reproductive piece. So all these things down here that have no reproductive pieces, those are blades or leaves. So you just go down to the base where it's the thickest, and we're going to get the width of the blades. And in your protocol, it tells you how many you need to get per plant, and it'd be, like I said, a very good thought to make sure that you confirm these widths with your lab group, because you have plenty of time to work on this stuff. It's important that we get accurate data. Now, spikes and inflorescence. So we're talking about, here's the comb here. We're going up to the top, and you see these spiky components. This is the reproductive parts. Now remember, these are all preserved, so they kind of dry and they lose color, and some of them keep more color than others. So as you're looking at all these different species, take into account a lot of the diversity that you're seeing. It's okay to write down colors and stuff, but colors are not always a good indication of a species, because environment can change how the plant looks, being in a drier environment than it is, or a drought condition, or if it had a little bit of disease to it. All that can affect color. But generally, the genes will allow the phenotype to be pretty much the same, say pretty much, for the given species. Right? So we're going to see a little bit of diversity within the species, but then we're also trying to see diversity between species so that we can actually do some phylogenetic studies based on their morphology, how they look. So in this picture here, we see an entire large amount of spikes, right? And this whole thing is called the inflorescence. So there's a couple things that we're going to take into account here. One is that the inflorescence is the whole thing, so we'd be looking at inflorescence, and one of the things you're going to record is the number of spikes. So you actually have to go in and count 1, 2, 3, 4, 5, 6, and then the question is this 7 and 8? We're going to have to look and use your probe and see if they come down to separate attachment points onto this main comb piece. And that's going to be not only a question for you, but your lab group, maybe a question for me, so that we can confirm, like I said, and get good, accurate data. But obviously, you're going to do some practice in class. I expect you to be responsible and getting good data. The other thing that each one of these spikes has is perigynia. These little pieces within the spike are called perigynia. So here's one pulled off, and inside the perigynia is going to be a keen, which is like a seed of the plant. So here we have a zoom in, and you're going to be doing a whole separate program, which I have a tutorial for. We're going to actually get data using digital pictures of the perigynia from one spike from your specific species. So lab aides have been working all summer to collect this data for you, and then you have to do all the measurements and enter it into our program that we're going to be working with. So this is one perigynia off of a spike from an inflorescence of one plant. So you're going to be getting the width of this perigynia, you're getting the length, you're going to get the area, and I'll show you how to do all that in the second tutorial. The other thing that you're going to be looking at is the akene. So this seed came out of this perigynia, and you're going to be looking at the width and lengths of the akenes. All this data is going to be compiled, and you're going to be able to have access not only to your data, but every other lab group's data. And the goal is that we're going to look to see if we can figure out if there's enough diversity between species and within species to get answers about lineage. And the cool part is that not only are you contributing to science, but we don't know the answer. So you're just as good of a hypothesis maker as we are. So keep that in mind, and like I said, if you have any questions, please make sure you ask. and follow your protocol because it's pretty descriptive. Thanks, and look for the second one on image J shortly.